Hello students, today we will be studying the histology of the salivary glands. Now in this video we will first be seeing the general structure of a gland, the duct and the acini, and then we will go on to study three important exam slides of the salivary glands. That's the serous gland, mucous gland and mixed salivary gland. Salivary glands. The oral cavity is surrounded by salivary glands such as parotid, submandibular and sublingual glands. Salivary glands secrete saliva in the oral cavity. These can be classified as major and minor salivary glands. Major salivary glands open in the oral cavity with the help of long ducts, and examples are parotid, submandibular and sublingual glands. The minor salivary glands are small glands located in the lamina propria of lips, cheeks, soft palate and tongue. Each gland has a duct and a secretory unit or acini. Salivary gland consists of highly branched duct system and a well-developed secretory acinar unit. Acini are lined by simple epithelium. Based on the nature of the secretory cells, the acini are classified as serous, mucous and mixed acini. Glandular tissue is surrounded by connective tissue capsule. Septa arising from the capsule divide the gland into number of lobes and lobules, which are separated in turn by interlobar and intralobar or intralobular connective tissue. Salivary ducts. Main salivary duct of the gland divides to form lobar, interlobar and intralobular ducts. Intralobular ducts are of two types, striated and intercalated ducts. Intercalated duct is lined by low cuboidal epithelium. Nucleus is pushed towards the base of the cell with apical cytoplasmic granules. These ducts connect the secretory acini with larger ducts, and these are predominantly seen in the serous glands. This here is a picture showing you the structure of an intercalated duct lined by low cuboidal cells with nucleus pushed towards the base of the cell. Striated ducts are lined by simple columnar epithelium. The lining cells show basal infoldings of plasma membrane, which gives striated appearance on H&E staining. Hence, they are called striated ducts. They connect intercalated ducts with interlobular ducts, and these are predominantly present in the serous glands. They are absent in the mucous glands. And this here is a picture showing us a striated duct lined by columnar cells with appearance of striations. Excretory ducts include interlobular, lobar and main excretory ducts. These ducts show gradual epithelial transformation from simple columnar to stratified cuboidal or stratified columnar epithelium. Main excretory ducts open into the oral cavity. This here shows us picture of excretory duct and the transformation from simple columnar to stratified cuboidal and then on to stratified columnar epithelium. We now go on to study the secretory acini. Salivary glands are tubulo-alveolar glands and have serous, mucous and mixed acini. Let us now see serous acini. These acini are lined by pyramid-shaped simple columnar epithelium with a round basal nucleus, large amount of rough endoplasmic reticulum in the basal region of cells that gives basal basophilia. The cells also show a well-developed Golgi complex and many secretory vesicles that is the zymogen granules, in the apical region of the cells that gives apical eosinophilia. Serous acinus has a very small lumen as the apices of the pyramidal cells occupy the central part of the acinus. So there is hardly any lumen seen. Example is parotid gland. This here is a picture of a single serous acinus where we see that it is lined by pyramid-shaped columnar cells. The apices jutting into the center. Hence, there is either no lumen appreciated or a very small lumen that is seen. Rounded nucleus in the center of the cell with basal basophilia and apical eosinophilia. Also seen here is a myoepithelial cell. Mucous acini. These acini are lined by pyramid-shaped simple columnar epithelium having a flat, elongated, basally placed nucleus and clear cytoplasm. The mucus secreted and stored in the cell in the form of mucinogen granules is lost during tissue preparation, thus giving an appearance of clear cytoplasm. Mucous acinus has a well-defined wide lumen, example being sublingual salivary gland. This here is a picture of a mucous acinus where we can see that it is lined by pyramid-shaped columnar cells. Flat, basally placed nucleus, clear cytoplasm, and a wide, well-defined lumen. Also seen here is a myoepithelial cell. Mixed acini. These are mucous acini with serous demilunes. Mucous acinus is capped with serous cells that form the serous demilunes of Guillain-Barré. Demilune is a half-moon shaped cap over the mucous acinus that consists of a few serous cells. All the serous cells of the serous demilunes pour their secretions in the lumen of the mucous acinus through fine canaliculi. Example, submandibular salivary glands. And this here is a picture of a mucous acinus capped with serous demilunes. This here is a mucous acinus with a myoepithelial cell, and these here are a few serous cells that cap the mucous acinus to form the serous demilune. Let us now see differences between serous cell and a mucous cell. With regards to the shape and size of the lining cells, serous cells are small pyramid-shaped columnar cells. Mucous cells are large pyramid-shaped cuboidal or low columnar cells. With H&E staining, serous cells show basal basophilia and apical eosinophilia. Mucous cells show clear cytoplasm because the mucus stored inside is washed away during tissue processing. The nucleus of a serous cell is round and placed in the basal part of the cell. The nucleus of a mucous cell is flat and is placed towards the basement membrane. Secreted granules seen in the serous cells are zymogen granules. Mucinogen granules are seen in the mucous cells. Let us see differences between a serous acinus and a mucous acinus. With regards to the size of the acinus, serous acini are smaller as compared to the mucous acini, while mucous acinus is larger as compared to serous acinus. Type of acini. Serous acini are compound alveolar type. Mucous acini are compound tubular or compound tubulo-alveolar type. The lumen is very small or hardly appreciated in the serous acinus. Mucous acinus has a wide, well-defined lumen. Cells. Serous cells are seen in serous acini and mucous cells are seen in the mucous acini. Secretions. Serous acini have thin, watery, enzyme-rich secretion. Mucous acini shows thick, mucoid secretions. Examples where we see serous acini is parotid gland. Mucous acini are seen in the sublingual salivary gland. This here is a picture to compare the serous acinus with a mucous acinus. We now go on to study the myoepithelial cells. These are also called as basket cells or myoepithelium. These are star-shaped cells that are present between the epithelium and basal lamina of an acinus. They have actin filaments in their cytoplasm. Their contractions help the acini to expel out their secretions. We now go on to study the three important slides. Histology of parotid gland. This is a serous salivary gland. It's the largest salivary gland in the body. Covered by a thick capsule and divided into a number of lobes and lobules. Main excretory duct or the Stenson duct opens into the vestibule of the mouth opposite the upper second molar tooth. Acini here. The parotid gland predominantly consists of serous acini. These acini are surrounded by myoepithelial cells. Duct C. Parotid gland has a number of intralobular, that is, intercalated and striated ducts, and also interlobular and lobar ducts. Intercalated ducts are lined by low cuboidal epithelium. Striated ducts are lined by simple columnar epithelium with basal infoldings. Other ducts are lined by stratified cuboidal to stratified squamous epithelium. In between acini, adipose tissue is also present. This here is a hand-drawn diagram of the parotid gland. What we see here are predominantly serous acini that are seen. We also see striated duct, interlobular duct, and intercalated duct. The connective tissue in between shows presence of blood vessels and adipose cells. What we see here is a low power view of the slide of a parotid gland. What we see here are predominantly serous acini along with the ducts and a few adipose cells in the connective tissue. A hyper view of the same shows the serous acini with hardly any lumen seen, and we also see here the interlobular duct and adipose tissue. Another hyper view showing a striated duct where we can clearly see the basal infoldings that give rise to the striations. This here is an aperio imagescope picture of the parotid gland showing us the serous acini with basal basophilia and apical eosinophilia, a rounded centrally placed nucleus. We also see an intercalated duct in the connective tissue along with a few adipocytes. Salient features for spotting for a slide of parotid gland will be presence of serous acini with narrow lumen, presence of serous cells having centrally placed round nucleus. We now go on to study histology of the submandibular gland. This is a mixed salivary gland. It has a well-defined capsule. Septa arising from the capsule divide the gland into a number of lobes and lobules. Acini seen here, the gland shows both serous and mucous acini, and a few mucous acini also show serous demilunes. Ducts. Gland has lobar, interlobular and intralobular, that is intercalated and striated ducts. Intercalated ducts are less in number as compared to those seen in the parotid gland. We see a hand-drawn picture of the submandibular gland where we see serous acini, mucous acini, and a few mucous acini with serous demilunes. So this here is a serous acinus. These are mucous acini, and what we see here are mucous acini with serous demilunes. Also seen in the connective tissue is interlobular duct, striated duct, and blood vessels and adipocytes. This here is a low power view of a slide of the submandibular gland. We can appreciate here serous acini. These are the mucous acini, and we also see a mucous acinus with a few serous cells, and that is the serous demilunes of Giannuzzi. A high power view of the same showing us serous acini, mucous acini, and a few mucous acini with serous demilunes. So when we see a mixture of serous acini and mucous acini, we should identify the slide as a slide of mixed salivary gland. Example being the submandibular salivary gland. This is an image scope picture of a mixed salivary gland showing us a clear view of the serous acini, the mucous acini, and what we see here is a mucous acinus with serous demilunes. Clearly seen here. Also seen here are the ducts in the connective tissue. Another duct seen here along with the adipose cells. Salient features for spotting for a slide of submandibular gland will be presence of both serous and mucous acini and presence of serous demilunes of Giannuzzi. We now go on to study the third slide, that is histology of sublingual gland. This is a mucous salivary gland. These are small masses of glandular tissue that lie in the substance of the tongue. The gland is not surrounded by a well-defined capsule. Acini. Gland has predominantly mucous acini. Few serous and seromucous acini are also present. Mucous secretory units are tubular. They are not purely acinar type. Ducts seen here. Glands do not have intercalated ducts. They have very few striated ducts. Interlobular and lobar ducts are present. A hand-drawn picture of the mucous salivary gland. What we see here are predominantly seen mucous acini. A very few serous acini are seen and very few mucous acini with serous demilunes are seen, but predominantly what we see are mucous acini. Also seen here are the striated ducts, connective tissue with adipose cells and blood vessels. A low power view of a slide of mucous salivary gland. What we see here are predominantly mucous acini having a clear cytoplasm, well-defined lumen, few blood vessels and ducts seen in the connective tissue, and very few serous acini seen. A high power view of the same. All these seen here as large acini with clear cytoplasm are mucous acini. Very few serous cells are seen along with the blood vessels and connective tissue. So when we see predominantly mucous acini with clear cytoplasm, white lumen, we identify the slide as a mucous salivary gland. An image scope image of a mucous salivary gland. We can see there are so many mucous acini seen, with hardly any serous acinus seen here. We see the interlobular connective tissue. So this we identify as a mucous salivary gland, a duct seen here. Salient features for spotting for a slide of sublingual gland will be presence of mucous acini with well-defined large lumen, presence of mucous cells having flat nucleus placed near the base of the cell. Thus, we have studied the basic features of the gland, the different ducts, and the different types of acini, and then we go on to study the three important slides here, that is histology of a serous gland, that is parotid gland, mixed salivary gland, that is submandibular gland, and mucous salivary gland, that is the sublingual gland. For further reference, you can go through Sontak K. Yogesh, Textbook of Human Histology by CBS Publishers and Distributors Private Limited, New Delhi, 2020, page numbers 189 to 195. Thank you.